Dear students, Garrett investigated many disorders that were related to families and generations, and that were moving from generation to generation because of certain kind of mutation or certain kind of changes that occurred in the genetic makeup or in the hereditary information that was present inside the ancestors of the affected families. Among those diseases was Alkaptonuria. Now let us see what is the disease. I will describe the condition in normal people as well as in the people with the disorder. First of all, let me tell you that normally our urine contains an acid that is known as homogenization acid. Now what happens in the normal people? In the normal people, this homogenization acid is converted, or you can say broken down into simpler substances. And the enzyme which is responsible for this whole reaction is homogenization acid oxidase. So this enzyme converts acid in simpler substance in the normal person, while in the people with alkaptonuria there is no enzyme. So homogenization acid goes as such in the urine, and when it is exposed to air, it is oxidized, and hence urine turns black. So by observing this whole phenomena, Garrett concluded that the patients who are suffering from alkaptonuria, basically they lack this enzyme, certain enzyme. And since the disease was running in the families, it means something happened to the DNA or the genetic information. So they concluded that when patients have genetic diseases, they lack certain enzyme which is required for normal functioning. So we can infer, or we can conclude from Garrett's finding, that information which is encoded within the DNA acts to specify particular enzyme. It means that here we have the cell, the nucleus, the chromosome. When you observe a chromosome, you see the DNA molecule, and a section of DNA molecule is the gene, and this gene is encoding special polypeptide. Polypeptides give rise to proteins, mostly enzymes. So when the genetic makeup is disturbed, ultimately the formation of polypeptide and the formation of protein and that particular enzyme, this will be disturbed, and hence the patients will either have the defective enzyme or they will have entirely no enzyme. Now this information was saved until 1941, and after that a series of experiments were conducted by two scientists at Stanford University. So now let us see what were those experiments and what did they present it to us.